Hello everyone, Namaskar. Today we will discuss about extraction of bioactives and pigments from processing waste. The concept which will be covered in today's lecture include bioactives, their types and extraction methods, and extraction of bioactives from food processing wastes, major pigments in fruits and vegetables and their extraction from fruits and vegetable processing wastes. And in the end, we will take up a few case studies related to the fruits and vegetable byproducts and waste utilization. You know that in the earlier classes also we discussed that fruits and vegetables represent the simplest form of functional foods because they are rich in several bioactive components. Fruits containing polyphenols and carotenoids have been shown to have antioxidant and activities, and they diminish the risk of developing certain types of cancers. Major processing waste includes trimmings, peelings, stems, seeds, cells, even residues remaining of the extraction of juice, oil, starch and sugar, etc. So, these are a huge amount of waste and byproducts is generated by the particularly from the fruits and vegetable process industry. So, these wastes can be used for extraction of bioactive components, the extraction of pigments, etc., because they contain huge amount of these valuable materials. And the recovered biomolecules and the byproducts can be used to produce functional ingredients and food ingredients, or they can be used in medicinal and pharmaceutical preparations. Okay. So, the fruits and vegetables, their bioactives etc. from the waste, they can be utilized in various products. That you should know that these fruits and vegetables, when they are processed into value added product, juice, nectar, sauces, dehydrated products, etc., they generate, as I told you, various waste or byproducts like pomace, seeds, rind in skin, peel, etc. So, these byproducts they can be subjected to appropriate technologies. For example, they can be used subjected to extraction technology either by conventional method or by novel extraction techniques, and the bioactives like phenolic, anthocyanins, flavonoids, tannins, etc., they can be extracted from them, and these extracted bioactives they can further be encapsulated so that their stability etc. during processing operations is ensured as it is improved. So, the encapsulation methods may be spray drying, ultrasonic emulsification, spray coating, fluidized bed coating and so on, even freeze drying. So, these encapsulated bioactives they can be used in the various functional food preparation, health food preparations as in ingredients in ice cream, crackers, cookies, cheese and what not. Okay. And the remaining after the extraction, whatever residue is remaining, that is waste, it is again a fiber rich component and it can be used for appropriately for such other purposes, which we will discuss little later either in biofertilizer or some other usage. Okay. So, this slide presents to you that bioactives, major bioactives present in fruits and vegetables and their function, particularly the fruits and vegetables waste. So, you see for example, tomato, when the tomato is processed for juice or ketchup or such other things pulp, then it peel, seeds and pomace, they are finally they emerge as a waste. So, they contain substantial amount of lycopene and essential lipids, and these lycopenes, essential lipids etc., they have very good anti-cancer agent. They reduce degenerative diseases, promote heart health. Potato peel, it contains valenic acid, gallic acid, caffeic acid, steroids, alkaloids etc., and which have again various health promoting functions like antipathogenic, anti-inflammatory, immune stimulation etc. Carrot peel and pomace contain phenols, beta carotene, pectin, which has antibacterial activity, they are also anti-cancer agent. Even pumpkin seeds, skins, cells, they have para-hydroxybenzoic acid, para-coumaric acid, they are antimicrobial. Okay. Even the apple pomace and peel, they contain epicatechin, anthocyanins, quercetin, okay, or other glycosides, and which helps in reduction of blood pressure. It has anti-inflammatory properties, etc. Grape skin seeds and stem, they have caffeic acid, ferulic acid, coumaric acid, chlorogenic acid, etc. Okay, and proanthocyanins, and they again have various health promoting activities, like they are cardioprotective, anti-aging agent, anti-cancer, they have enhancement of gut health, and so on. So, all these pomegranate seeds, citrus fruit, even mango, they are waste, that is whether seed, peel, pericarp, kernel, pulp, they have good valuable components. Like mango kernel and peel, they contain anthocyanins, ellagic acid, quercetin, tannins, mangiferin, etc., which have hypercholesterol, the extension of IL stability, they can be used, they have anti-cancer, antimicrobial activities, so on. So, the various these bioactives from the waste, they can be extracted either using novel technologies or conventional technologies. Okay. Novel technologies include supercritical fluid extraction, subcritical water extraction, ultrasound assisted, microwave assisted, or PEF assisted techniques. Okay. Conventional technology include hydrodistillation, solvent extraction, maceration, and so on. And the details of all these technologies, extraction methods, equipment used, operating parameters, etc., we had earlier discussed in earlier classes. So, I will skip this today, but the thing which I want to communicate is that these technologies appropriately, of course, since the raw material is changed, that it is a based, etc. So, sometime the adjustment of the process parameters, process conditions might be required, but the same technology can be used, and then extracted materials can be applied as used as an ingredient in food processing. So, let us now discuss about the pigments, that is pigments which are present in fruits and vegetables. They are natural colors, okay. They are good source of, and the fruits and vegetable processing waste, they are again very good source of food color. Okay. And this natural food color is any dye, pigment, or any other substance obtained from fruits, vegetable. You can see they have the multicolored, all these red, blue, green, yellow, etc. all these colors, they are because of the presence of various organic compounds which are known as pigments, and they contribute to the color of this commodity. So, the color come from variety of sources, like even seed, some fruits, their peel, vegetable, even algae, insect, grass, all these things, they contain these coloring components, and these coloring components you can see here in the picture. These various part, either seed can be taken in some fruit, pulp can be taken, peel can be taken, and it can be extracted, that is the color can be extracted from these components. Okay. So, the fruits and vegetables, that is the they contain natural colors of the in the form of 4 major groups of compounds. These are either yellow, orange, and red colors carotenoids, red, blue, purple anthocyanins, green chlorophylls, and red betannins. So, these are the 4 anthocyanins, carotenoids, chlorophyll, and betannins. Betannins, that is the major group of colored compound or pigments present in most of the fruits and vegetables. Okay. So, we will discuss their characteristic chemical nature briefly one by one. The carotenoids, they are tetraterpene pigments which exhibit yellow, orange, red, and purple colors. Carotenoids typically contain only carbon and hydrogen. So, they are basically hydrocarbons, and particularly they belong to the subclass of unsaturated hydrocarbon, because you can see here in the structure they have high degree of unsaturated bonds, and it is these unsaturated bonds, that is the compound, they absorb the colors of different intensity, different hue, these unsaturated bonds, and they accordingly they are colored compounds. Carotenoids with molecules containing oxygen such as lutein and zeaxanthin are called xanthophylls, and in many fruits the color is present in the form of xanthophyll. The unoxygenated, that is oxygen free carotenoids such as beta carotene, alpha carotene, lycopene, they are all known as carotenoid or carotenes. Okay. So the carotenes, they are a precursor of vitamin A, that is they are called provitamin A, that is when these carotenoids, that is most of these carotenes which are present in plant foods, they have the provitamin A activity like beta carotene, alpha carotene, beta cryptoxanthin and so on. So, the provitamin A means when it is consumed in body, these are converted into vitamin A. So, that is why they are precursor of vitamin A, very valuable component for our normal health. And also there are certain carotenoids like lutein, zeaxanthin, lycopene, etc. Lycopene is the color of the tomato. So, they do not have any provitamin A activity. So, they are known as non-provitamin A carotenoids. Okay. So, here in the slide, that is there are the carotenoids type and their major sources like alpha carotene is found in carrots, pumpkin, winter squash, plantain, collard greens, etc. Beta carotene major sources include carrot, leafy greens, sweet potato, pumpkin. Okay. Lutein and zeaxanthin are found in leafy greens, summer winter squash, Bruce spouse fruit, yellow corn, and even beta cryptoxanthin is found in pumpkin, papaya, sweet pepper, orange, carrot and so on. And here the extraction pattern, you can see that absorbance, they have these compounds like carotenes, they have the absorption spectra somewhere around 475 or 80, that is a 450 around they get the peak, okay, at 450 wavelength, whereas the chlorophyll A and chlorophyll B, they have somewhere around in between 600 to 700 nanometer. So, they have these various compounds, they have different, they absorb absorption spectra at different wavelength, they get the peak, and that is why how they are normally assessed or characterized. Okay. So, these carotenoids, they are very, very useful against the various chronic diseases. For example, these the carotenoids which they have antioxidant, they provitamin A activities, they have blue light absorption effect, and that is a beneficial against the macular degeneration, cataract, retinoid pigmentosa, etc. So, in the eye diseases, these carotenoids have play important role. Similarly, in the skin diseases, that is the this antioxidant property or anti-inflammatory property, or they lower down the UV light mediated damages, they give useful effects are that is in the skin sunburn, they reduce melanoma, etc. is reduced. Similarly, in the cancers or metabolic syndrome, etc., these carotenoids have. So, the carotenoids are very major class of compounds. Okay. So, they have their full potential in the whole fruits and vegetables. That is when we take raw fruits and vegetables, we get the carotenoid, but many of these, their health value, in fact, when they are little bit lukewarm, heated, etc. So, their health value is health potential or potential to fight against the diseases is improved, is increased. Okay. Then anthocyanins, that is another class of compound, they are colored water soluble pigments belonging to the phenolic group in glycosylated forms. Anthocyanins are responsible for red, purple and blue color in fruits and vegetables. Berries, currants, grapes and even some tropical fruits have high anthocyanin content. Red, they have red to purplish blue color. Even leafy vegetables, root, tubers are the edible sources that contain a high value of high level of anthocyanins. Among the anthocyanin pigment, cycladine 3 glycoside is the major compound found in most of these plants. Okay. So, here these are the major groups of anthocyanin like cyanidin, delphinidin, malvidin, paraxetin and pionidin, and their sources like malvidin is found in the flowers, red wine, blueberry, cyanidin is found in the grapes, blackberry, blueberry, cranberry, raspberry, apples and so on. So, all these compounds, they can be they are used for various operations. So, these colors remain, some of them, the major portion of these remains in the peel or in the pomace, etc. So, they can be used to extract. So, the color and stability of these carotenoids are influenced by pH, light, temperature and structure. In acidic condition, anthocyanins appear as red, but they turn blue when the pH increases. So, the particularly color nature of these carotenoids, etc. may be influenced during processing, but the health value may of course remain intact, or it may in some cases may get improved. Okay. This is the structure of major anthocyanins, which we saw in the last PPT. So, they are the structure, they are basically mostly you see phenolic compounds, etc. They have benzene ring in the structure, they are highly unsaturated, okay. And there are little difference in the structure compound one and each other. So, because of this they sometimes have different colors, intensity, color hue, and also their health value. Okay. Then the health benefits of the anthocyanins include that they have the antioxidative activity, antimicrobial activity, they improve visual and nutraceutical and neurological health, they protect against various non-communicable diseases. Okay. Chlorophyll, these are another major color compound, the pigment, that is green color compound in most of the plants, green color plants and leaves, etc. They are porphyrins, which are this macrocyclic tetrapyrrole pigments, in which the pyrrole rings are joined by methyl bridge and the double bond systems form a closed conjugated loop, and there is a centrally located magnesium. These all pyrrole rings, they are joined by the magnesium atom and the coordinated with the nitrogen. Okay. There are two types of chlorophyll, that is chlorophyll A as well as chlorophyll B. Chlorophyll A and B are both the typical green pigments of higher plants. They occur in various proportion, like for example, they are found in the ratio of 3:1 in lettuce leaf, or in the ratio of 1:2 or 1:4 in different varieties of tree tomato fruits. Chlorophyll A appears blue green in color, whereas the chlorophyll B is yellow green. Okay. Chlorophyll A has a methyl group and the chlorophyll B has a formyl group, that is the R. This R group which you see here in the structure, this is the structure of chlorophyll. So, R group in the A is methyl and in the B is a formyl group, and both they have, as you can see in the structure, they have an ethyl group at the carbon position 2, C2 and C4 positions respectively, and there is a carboxymethyl group at the C10 position of an isocyclic ring, and a phytyl group, which is here in the last is the phytyl group, okay, and it is esterified to the propionate at the C7 position, and it is actually the phytyl group. When the green chlorophyll containing vegetables, etc. are processed, sometimes these phytyl group is removed or central magnesium is removed, and accordingly that chlorophylls are converted into pheophytins or pheopharbides, etc., and their color may be little bit changed. So, particularly the green vegetables, when they are during cooking process and during processing, they undergo subject that changes into their color hue. Okay. The green color of vegetable or fruits is due to the presence of, as I told you, chlorophyll, okay, and it is affected by aging, enzymes, weak acid, oxygen, heat and light. The degradation of chlorophyll colors occurs during ripening of fruits, senescence of green vegetables, and thermal processing of foods. Okay. And this chlorophyll B is reported to be thermally more stable than the chlorophyll A, and these both form of chlorophyll, they have immense health benefits, like they help in weight loss, they smoothen the digestive system, act as antioxidant, relieve swelling, they protect DNA against fried foods, etc., and they have heal the injuries process, wounds and injuries. Betannins, betannin is a red glycosidic food dye obtained from the beet roots. It is a betallin pigment together with isobetanin, probetan and neobetanine. Other pigments contained in the beet are indigoxanthin and bulgioxanthin, and the color of betalain pigment depends upon the pH. They have various color in this, even in the many flowers, etc. These betalain, the chemical structure of the betalain is shown here. Okay, so when in the pH changes, the color it changes. That is between the pH 4 and 5. It is bright bluish red color. It is blue violet as the pH increases, and as the pH reaches alkaline level, it degrades by hydrolysis, and its yellow to brown color hue is obtained. Okay. Sources of vitamins are beet juice, as I told you, aptamina, Swiss chard, amaranth, etc. They have various health benefits, like they are anti-diabetic, they reduce LDL, they reduce blood pressure, and so on. Okay. So, the applications of this natural color in the food systems, like these colors you have seen natural, that is anthocyanin, phycocyanins, chlorophylls, carotenoids, betalains. These are the major groups of the coloring compounds present in the various plant material and in the waste also. So, they can be accordingly used, like betalains, etc. They can be used in the manufacture of yogurt, beverages, ice cream, confections, cake mixes, etc. In the anthocyanin, they may be better incorporated into bakery product, dairy product, frozen desserts, beverages, phycocyanins in gums, confections, soft drinks, dairy. So, all these, there are various like carotenoids, they can be used in meat products, in the butter and vegetable oils, cheese, paste, and so on. Chlorophylls, that they can be used in the beverage mixtures, yogurt, puddings, ice creams. So, they have lot of ideas. They can be incorporated into the various food products, and in fact, they have a good potential to substitute, to replace the synthetic dye. Okay. So, utilization of now tomato processing byproducts. Okay. So, the tomato, when it is processed for juice, concentrate, puri, etc., then you get the tomato pomace, and this tomato pomace can be used for production of biomethanol or biopolymers production, okay, or even that is hydraulic enzyme production can also be used, like tomato peels and tomato seeds. So, there are these pomace, and this peel can be used for bioactive recovery, that is these bioactive can be used in pharmaceutical, and this from seed, also seeds, seed oil extraction, and this both seed oil residue remaining of the seed oil extractions, that is solid residue, it can be then dye removal, etc. So, various uses are there. So, in fact, bioactives, phytochemicals like sterols, tocopherols, carotene, terpenes, they are all present in the tomato byproducts, and they have good antioxidant potential. Okay. It can be, that is the lycopene, as I told you, it is the major color pigment found in the tomato. So, after that extraction of the juice, this color, majority of it is retained there in the peel or in the pulp, what is it do. So, this after the processing, that is a tomato waste which is get, it can be used, there is same to either supercritical fluid extraction or such other extraction methods. Okay, and the color can be extracted. This can be used either both for nutraceuticals as well as after the extraction of color, the value material, they can be also used in the cosmetic products, etc. Okay. So, here in this figure, it is shown that SFE extraction kinetics of oil from tomato waste at 500 bar and at 60 degrees Celsius temperature. So, as S2F ratio, that is the mass ratio of solvent to feed, as it increases, okay, that is extraction efficiency, that is accumulated yield of the lycopene is increased. So, here almost you can see that this it has reached after that, so from 0 to 5 solvent extraction, that is increasing very fast, and after that it is almost stabilized. So, accordingly this can be used, that yes, what should be the solvent, what should be the feed, what is proper ratio, so that you can get better recovery. Okay. So, this is a procedure we have taken from the literature. Okay, source is given here. They have recommended that the tomato industrial byproducts collect and give a pre-treatment, you get tomato seed and peel, and the peel can be subjected with the refined olive oil, RO, then macerated and extracted okay, and then the TP and RO position may be various band and box design, that is these parameters of the TPP and RO level, temperature and even agitation speed, various combination and permutation in these ranges are used, and then finally the process parameters are optimized for better recovery and yield of the this lycopene extraction, and they have been some parameter it as about as almost 100% recovery they have reported. Okay. So, it is and the quantification of lycopene can be done by HPLC. These are the structure of lycopene and beta carotene. You see that they both have the almost similar structure, except here you see in the case of lycopene this is open, wherein the beta carotene it is closed, and because of this you find the difference, huge color difference in the lycopene and beta carotene. Okay. Then the here that is some researchers they have reported, there is a recent research report of 2022 only, that they have compared the supercritical carbon dioxide extracted oleoresin from the tomato seed and peel and the hexane extracted oleoresin. Okay. And they assessed is the bioaccessibility of the lycopene, bioaccessibility of the lycopene, and it was found very interesting information that the supercritical, that is carbon dioxide extraction, it is much biologically accessible. It has much biological accessibility in compared to the hexane extracted oleoresin. Okay. This is under comparison of extraction methods, all right, that is the extraction sample, what is the method used and what is the carotenoid obtained, like from tomato peel the enzyme extracted assisted extraction you get 356 mg per 100 gram of dry weight. So, accordingly of course, that is the using you see when the novel extraction technologies are used, probably they give better yield. Okay. So, from the dried tomato peel or tomato waste or mixture of peel and seeds, even dried tomato pomace. So, different component, tomato skin and seed and tomato even dried samples with the skin and seeds. So, various combination and permutations are used, and even different extraction methods are you get in the some cases. In fact, this tomato dried tomato pomace as well as the tomato waste as such and tomato peels. From there you get a good recovery of a good amount of lycopene. Okay. These are the technology again reported in the literature for the utilization of orange processing byproduct, that is for the extraction of pectin and extraction of essential oil. For pectin, that is orange peel is converted into powder using appropriate technology, then it is treated with 0.05 normal and HCl boiled for at 100 degrees Celsius for 1 hour and cooled to room temperature. After that it is filtered and the solid residue filtrate is separated. The portions the liquid is obtained or the residue which is obtained, it is treated with alcohol, separate precipitate, dried, and you get the pectin powder, pectin. Okay, it can be further purified and characterized. Okay. Essential oils, that is orange peel, without the flavonoid is taken, it is saturated in calcium hydroxide solution 1% for 8 hours, and then this removed from the solution. Okay. Dried at 45 degrees Celsius, powdered, and then this powder is distilled using either Saxelate apparatus or such other apparatus, and that is 80 gram powder plus 500 ml water and 10 gram sodium chloride. They have get oil, and this add sodium sulphate, filter, add chlorine that is ethyl chloride, CH2Cl2, concentrate, and store. So, you get extraction, it can be essential oil is extracted. Okay. Here is a schematic of the utilization of potato processing byproduct, that is potato when it is processed for preparation of chips or for preparation of other products. It gives that is the peel which we obtain. The peel may be source of dietary fiber, it is rich in phytochemicals or it is rich in various bioactives, it is beneficial to human health. Okay. So, that is the various products that they can be further utilized in it discussed earlier. Like potato peel, it is dried and made into powder, then it is extracted, and the potato peel extract, it is can be used in various beverages or such other products. Okay. Then here it is the extraction of useful compound from potato peel by sequential hydrothermal treatment. They have given the potato peel slurry. Okay, it is two, that is the treated peel. So it is stage 1 and stage 2. That is stage 1 directly potato peel slurry is given the crude extract, or it is further treated. Okay, and then you get treated stage 2. So crude stage 1 and crude stage 2, they can be further given that sugar precipitation and polysaccharides are separated. Then alkaline precipitation, glycoalkalides 1 and glycoalkalides 2 are separated, and finally the purification. By purification you get polyphenols 1 and polyphenol 2 and the other nutrients, etc. Of course, the author has given, so there are some there. If you are interested, you can further read to this paper and get more about it. But the thing which I am trying to tell here is that yes, using appropriate method, appropriate processing technology and process parameters, these valuable components with having specific characteristics, they can be obtained from these peels and waste. So here even extraction of phenolics from potato peel by conventional, that is UNV, which is here, and microwave assisted and ultrasound assisted methods, and there is a comparison is shown here, and you can see here very interesting application when this it is ultrasound and microwave assisted. Particularly you see the conventional have a solvent extraction and the conventional GLA plus 5% alcohol. So very low when it is used, that is only that is conventionally extracted. But when the same technology is used in combination with either microwave or combination with ultrasound, the yield is substantially increased in both, which was in the single solvent extraction was less, that when it is combined, it is quantity is increased. Okay. So this is better, that is you go for either microwave assisted extraction or ultrasound assisted extraction. Okay. Then finally, I will summarize that yes, that these fruits and vegetables, when they are processed, they generate a huge amount of waste stream or byproducts, and all these waste and byproduct almost in some cases, if I say that almost it is 50%, almost 50% goes as waste, or 50 to 60% some cases. Okay. So that waste is a very valuable source. So if this waste is left unattended, this will create environmental problems, so many problems. But if there is a proper mechanism in place, okay, for their collection, and these are even the food processing industry, like for example tomato processing industry, it is their responsibility. In fact, they should rather have some arrangement that whatever waste is being produced, it is properly treated, and even if they cannot use that, they should have some networking arrangement with that some startup and other companies who can utilize these into various value added product for extraction of color, for extraction of pigments, etc., in the preparation of various food industry, cosmetic industry, or pharmaceutical industry, etc. So a proper management, okay, of organic byproducts, it will furnish environmental as well as economic benefits by reducing the food waste, and the manufacturer, in fact, their economic advantage which will get, that will be always good, and environmental impact will be less, and health value products will be available, health ingredients. So always it is a win-win situation, but the one has to take care that yes, we should have a proper approach for utilization of these valuable byproducts or waste products of horticultural and plantation crop industry. These are the references used in this lecture. Yes, thank you very much.